Which among the following made it possible the cutting of DNA at specific locations? The correct answer is option B, restriction enzymes. The restriction enzymes act as molecular scissors that recognize and cut the DNA at specific sequences of bases. And they are naturally found in bacteria and serve as a defense mechanism against viral DNA. So, restriction enzymes have become essential tools in molecular biology, particularly in genetic engineering, where they are used to cut the DNA for cloning, sequencing, and various other techniques. So, the answer is option B, restriction enzymes. Whereas coming to option A, ligases, these are the enzymes that join, that is by the process of ligation, two pieces of DNA by forming phosphodiester bonds. So option A is incorrect. Option C, probes. Probes are short labeled DNA or RNA sequences which is used to detect the presence of complementary sequences in a sample. So option C is incorrect. Option D, selectable markers. Selectable markers are the genes that are used to identify the cells that have taken up a foreign gene, typically by providing a resistance to the antibiotics. So option D is also incorrect. The correct answer is option B.